RNA processing. Eukaryotic mRNA precursors are processed by 5' capping, 3' cleavage, polyadenylation, and RNA splicing to remove introns before being transported to the cytoplasm where they are translated by ribosomes. Next, we are going to learn about RNA processing. So, eukaryotic mRNA precursors are processed by 5' capping, 3' cleavage or polyadenylation, and RNA splicing to remove introns before being transported to the cytoplasm where they are translated by ribosomes. 5' capping. Eukaryotic mRNA have a peculiar enzymatically dependent cap structure consisting of 7-methyl guanosine residue joined by a 5' triphosphate bridge. During transcription, 7-methyl guanosine is added to the 5' end of nascent mRNA. The initial stages in RNA capping are catalyzed by a dimeric capping enzyme which associates with the phosphorylated carboxyl terminal tail domain CTD of the RNA polymerase II. Then, the reactions that cap the 5' end of each RNA molecule synthesized by the RNA polymerase II. The final cap contains a novel 5' to 5' linkage between the positively charged 7-methyl G residue and the 5' end of the RNA transcript. The final cap contains a novel 5' to 5' linkage between two positively charged 7-methyl G residue and the 5' end of the RNA transcript. The letter N represents any one of the 4 ribonucleotides, although the nucleotide that starts an RNA chain is usually a purine. So, 5' end of nascent RNA, GPPP plus PPNP, then guanylyl transferase, guanine 7-methyl transferase, and mapping. One subunit of the capping enzyme removes the gamma phosphate from the 5' end of the nascent RNA emerging from the surface of the RNA polymerase II. The other subunit transfers the GMP moiety from GTP to the 5' diphosphate of the nascent transcript, creating the guanosine 5' triphosphate structure. In the final step, separate enzyme transfer methyl groups from S-adenosyl methionine to the N7 position of the guanine and the 2 oxygens of ribose at the 5' end of the nascent RNA. If mRNA has only a methyl group on 7-guanine at the 5' end, then it is called cap 0. The cap 0 is a predominant cap in unicellular organisms. In some species, a methyl group is added to the second as well as third nucleoside of the capped mRNA with the methyl groups on the terminal 7-guanine and T2' OH position of the second nucleotide at the 5' end is known as cap 1. This is the predominant cap in the multicellular organism. Similarly, if methyl group present at both second and third nucleotides, then it is called cap 2. Cap 0, cap 1 and cap 2. Polyadenylation. Virtually all eukaryotic mRNAs have a series of up to 250 adenosines at their 3' end called poly A tail. This are not specified by the DNA and are added to the transcript by a template independent RNA polymerase called poly A polymerase. The polymerase does not act at the extreme 3' end of the transcript but at an internal site which is cleaved to create a new 3' end to which the poly A tail is added. The poly A tail appears to have several functions: export of mature mRNA from the nucleus, affect the stability of some mRNAs, and serve as a recognition signal for the ribosome. Virtually all eukaryotic mRNAs have a series of up to 250 adenosines at their 3' end called poly A tail. These are not specified by the DNA and are added to the transcript by a template independent RNA polymerase called poly A polymerase. This polymerase does not act at the extreme 3' end of the transcript but at an internal site which is cleaved to create a new 3' end to which the poly A tail is added. The poly A tail appears to have several functions: export of mature mRNA from the nucleus, affect the stability of some mRNAs, and serve as a recognition signal for the ribosome. A single processing complex undertakes both the cutting and polyadenylation. The signal is needed for both cleavage and polyadenylation. In mammals, polyadenylation is directed by a signal sequence in the mRNA, almost inaudibly given. This sequence is located between 10 and 30 nucleotides upstream of the polyadenylation site, which is often immediately after the dinucleated 5' CA 3' and is followed by 10 to 20 nucleotides later by GU-rich region. Both the poly A signal sequence and the GU-rich region are binding sites for multi-subunit protein complexes, which are respectively the cleavage and polyadenylation specificity factor and the cleavage stimulation factor. Generation of the proper 3' terminal structure requires an endonuclease consisting of the components CF5 cleavage factor 1 and CF2 cleavage factor 2 to cleave the RNA. So cleavage is followed by polyadenylation in a tightly coupled manner. The poly A polymerase has a non-specific catalytic activity. The polyadenylation reaction passes through two stages. First, a rather short oligo sequence is added to the 3' end pre-mRNA. Then assembly of the polyadenylation complex, polyadenylate binding protein, polyadenylation. Then the cleavage and polyadenylation of pre-mRNAs. Cleavage and polyadenylation specificity factor CPSF binds to the upstream poly A signal. CSTF interacts with the downstream GU or U-rich sequence. Binding of poly A polymerase PAP then stimulates cleavage and then adds residues at a slow rate to the 3' hydroxyl group generated by the cleavage reaction. In the second phase, the oligo A tail is extended to the full 200 residues length. This reaction requires another simultaneous factor that recognizes the oligo A tail. The additional factor requires polyadenylate binding protein or PADP, which helps the polymerase to add the adenosine, probably influences the length of the poly A tail that is synthesized and appears to play a role in maintenance of the tail after synthesis. So histone genes are unusual among eukaryotic genes in that they do not have introns, and histone mRNA do not have poly A tails. Furthermore, in almost all eukaryotes, genes are arranged in multiple tandem domains, each domain carrying one copy of each of the 5 histone genes. So just an explanation for this feature in terms of the special requirement for histone synthesis. Histones are required in very large amounts during only one brief period, the beginning of S phase in the cell cycle. Furthermore, they are always required in equivalent amounts to make nucleosomes. The presence of multiple copies aids in rapid transcription, and an equal number in each domain should help to maintain balance in production. The absence of introns and poly A tails means that two of the major steps involved in processing most eukaryotic genes are avoided so that histone mRNAs can be delivered rapidly to the cytoplasm. So just a means by which you could separate mRNAs from the other types of RNA in an eukaryotic cell. Attach an oligo DT or oligo U sequence to an inert support to create an affinity column. When RNA is passed through the column, only poly A containing RNA will be retained. Then intron splicing. Non-coding DNA also found within most eukaryotic genes. Such genes have a split structure in which segments of coding sequence called exons are separated by non-coding sequence or intervening sequences or introns. The entire gene is transcribed to yield a long RNA molecule, and the introns are then removed by splicing. So only exons are included in the mRNA. The process of excising the sequences in RNA that correspond to introns and joining the sequences corresponding to exons is called RNA splicing. Mechanism of RNA splicing varies depending on the types of introns. There are many types of pre-RNA introns. Two types, the GUAG and AUAC introns, are commonly found in eukaryotic protein coding genes. Eukaryotic nuclear pre-mRNA, eukaryotic nuclear pre-mRNA, eukaryotic nuclear pre-rRNA, organelle rRNA, some prokaryotic rRNA, then organelle rRNA, some prokaryotic rRNA. Group 3, pre-tRNA introns, eukaryotic nuclear pre-tRNA, then various RNAs also. So splicing of GUAG intron. In GUAG intron, the first two nucleotides of the intron sequence are 5' GU 3', 5' splice site or donor site, and the last two 5' AG 3' splice site or acceptor site. So pre-mRNA, exon, intron, exon portions, splice site, branch point, splice site, consensus sequence around 5' and 3' splice sites in pre-mRNA. A pyrimidine-rich region called polypyrimidine tract, not shown in figure, near the 3' end of intron is found in most cases. The branch point adenosine, also invariant, usually is 20 to 50 bases from the 3' splice site. AUAC intron is rare class of introns because the conserved 5' GU and 3' AG dinucleotides are replaced by AU and AC respectively. Then trans-esterification reactions. Splicing of GUAG intron involves two trans-esterification reactions. Cleavage of 5' splice site occurs by a first trans-esterification reaction promoted by the hydroxyl group attached to the 2' carbon of an adenosine nucleotide located within the intron sequence. The result of the hydroxyl attack is cleavage of the phosphodiester bond at the splicer site, 5' splice site, accompanied by formation of a new 5-2 phosphodiester bond linking the first nucleotide in the intron, the G of the 5' GU 3' motif, with the internal adenosine. This means that the intron has now been looped back on itself to create a Larian structure. So cleavage of the 3' splice site and joining of the exons results from second trans-esterification reaction. This one promoted by the 3' OH group attached to the end of the upstream exon. This group attaches the phosphodiester bond at the 3' splice site, cleaving it to and so releasing the intron as the Larian structure, which is subsequently converted back to the linear RNA and degraded. At the same time, the 3' end of the upstream exon joins to the newly formed 5' end of the downstream exon, complexing the splicing process. A large number of ATP is consumed during the splicing reaction. This energy is required not for chemistry but for assembly of splicing apparatus. Then first trans-esterification, second trans-esterification and Larian structure. Exon 1, first trans-esterification, second trans-esterification, Larian structure. So two trans-esterification reactions that result in splicing of exons in pre-mRNA. In the first reaction, the ester bond between the 5' phosphorus of the intron and the 3' oxygen of the exon 1 is exchanged for an ester bond with the 2' oxygen of the branch site A residue. In the second reaction, the ester bond between the 5' phosphorus of exon 2 and the 3' oxygen of the intron is exchanged for an ester bond with 3' oxygen of exon 1, releasing the intron as a Larian structure and joining the two exons. Then splicing apparatus. The central component of the splicing apparatus for GUAG introns are the snRNAs called U1, U2, U4 and U5 and U6. These short DNA molecules associated to proteins to form small nuclear ribonucleoproteins or snRNPs. The snRNPs together with other accessory proteins attached to the transcripts and form a series of complexes, and last one, which is the spliceosome, the structure within which the actual splicing reactions occur. The process of assembly of snRNPs and various protein factors occur as follows. The commitment complex, E complex, initiates the splicing activity. The complex comprises U1 snRNP, which binds to the 5' splice site partially by RNA base pairing. The splicing factor U2AF, which binds with the polypyrimidine tract and the 3' splice site, and members of a family called SR protein. Name SR proteins is given due to the presence of an ARG SR rich region. So SR protein interact with one another via their ARG SCR rich regions and also bind to RNA. Binding of U1 snRNP to the 5' splice site is the first step in splicing. The E complex is converted to an A complex when a U2 snRNP bind to the branch site. The pre-spliceosome complex A complex comprises the commitment complex plus U2 snRNP. The later attaches to the branch site. At this stage, an association between U1 snRNP and U2 snRNP bring the 5' splice site into close proximity with the branch point. The spliceosome is formed when U4U6 snRNP So a pre-spliceosome complex, A complex, comprises the commitment complex plus U2 snRNP. The later attaches to the branch site. At this stage, an association between U1 snRNP and SU2 snRNP brings the 5' splice site into close proximity with the branch point. The spliceosome is formed when U4U6 snRNP, a single snRNP contains 2 snRNAs, and 5U5 snRNP attached to the pre-spliceosome complex. Following formation of the A complex, and the other snRNPs involved in splicing associated with the complex in a defined order. The B1 complex is formed when U5 and U6 snRNPs bind to the A complex. This complex is regarded as a spliceosome since it contains the components needed for the splicing reaction. It is converted to the B2 complex after U1 is released. The catalytic reaction is triggered by the release of U4. This requires hydrolysis of ATP. When U4 dissociate with U6 snRNPs, U6 snRNA can pair with U2 snRNA to form a catalytic active site or C complex. This results in additional interactions that bring the 3' splice site close to the 5' site and the branch point. All three key positions in the intron are now in proximity to the transesterification occur as a linked reaction, possibly catalyzed by U6 snRNP, completing the splicing process. Spliceosome mediated RNA splicing mechanism. RNA splicing is catalyzed by a spliceosome formed from the assembly of U1, U2, U5 and U4 and U6 snRNP plus other components. After assembly of the spliceosomes, the reaction occurs in two steps. The step 1 is the branch point A nucleotide in intron sequence, which is located close to the 3' splice site, attacks the 5' splice site and cleaves it. The cut 5' end of the intron sequence thereby becomes covalently linked to its A nucleotide, forming the branched nucleotide. In step 2 and 3, 3' end of the OH end of the first exon sequence, which was created in the first step, adds to the beginning of the second exon sequence, cleaving the RNA molecule at the 3' splice site. The two exon sequences are thereby joined to each other and the intron sequence is released as a lariat. And next portion is alternative splicing. During RNA splicing, the introns are precisely excised and the exons are ligated together. The majority of nuclear pre-mRNAs are spliced constitutively, that is, only one mature mRNA species is generated from single pre-mRNA in all tissues. In some cases, however, alternate 5' and 3' splice sites are used during splicing, resulting in production of more than one mRNA species from a single pre-mRNA. The production of different RNA products from a single product by chance changes as the usual usage of splitting junctions in described as alternative splicing. Spliced mRNA troponin T exon, some other exons, introns, splicing will be happening. So alternative splicing in the troponin T gene shown here is a reason of this gene encoding 5 exons which generate two alternatively spliced forms as indicated. One contains exon 1, 2, 4 and 5. The other contains exons 1, 2, 4 and 5. 1, 2, 3 and 5. Alternate splicing has been documented for many eukaryotic genes. The utilization of alternative 5' or 3' splice sites can result in structurally distinct mRNAs by either excluding potential exon sequence or incorporating otherwise non-coding intron sequence. For some pre-mRNAs, alternative splicing is a non-regulated event. For others, the choice of alternative splice sites is regulated in a tissue-specific manner. So Drosophila sex determination provides the best example of regulated alternative splicing. In Drosophila, the primary signal for determining whether the fly develops as a male or female is the ratio of number of X chromosome and autosome set. Three crucial gene products are involved in transmitting information about this ratio. So the many other genes that specify male and female characteristics. The genes are called sex lethal, transformer and double sex. The function of this gene products is to transmit the information about the X chromosome or autosome ratio to the many other genes that are involved in creating the sex-related phenotypes. Number of X chromosomes, set of autosome, XL gene product, TRA gene product, DSX gene product, male or female fly. Sex determination in Drosophila, the gene products act as a sequential cascade to determine the sex of the fly according to the X chromosome or autosomal ratio. Then, required regulated alternative splicing during expression of genes involved in sex determination in Drosophila. The first gene that undergo for alternative splicing is XXL, whose transcript contains an optional exon which results in an inactive version of protein XXL. Then functional XXL protein in female blocks the 3' splice site on the first intron of the TRA pre-mRNA. This results in an mRNA that codes for functional tRNA protein. In males, there is no SXL, so the 3' splice site is not blocked and dysfunction mRNA is produced. tRNA pre-mRNA, male, female, no tRNA protein synthesis. Then SXL induces cryptic splice site selection in the pre-tRNA mRNA. tRNA pre-mRNA. The resulting female-specific tRNA protein is again involved in trans-activist splicing. tRNA protein interacts with splicing factors and binds with an exon of a third pre-mRNA DXX, products of DXX gene. Thus result in female-specific DSX protein. The two versions of DXX are the primary determinants of Drosophila sex. This XXL is a precursor that acts negatively to block a splice, where the tRNA protein are activators that act positively to induce a splice. Then gene modified definition. The definition of a gene has had to be modified since the discovery of alternative RNA splicing. The work on Neurospora showed that most genes correspond to a region of genome that directs the synthesis of a single enzyme. This leads to the hypothesis that one gene encodes one polypeptide gene, but it is now clear that many DNA sequences in higher eukaryotic cells produce two or more distinct polypeptides by means of alternative RNA splicing. How then is a gene to be defined? According to modified definition, a gene is a DNA sequence that is transcribed as a single unit and encodes one set of closely related polypeptide genes. This definition of gene also include those DNA sequences that encode protein variants produced by post-transcriptional process other than RNA splicing, such as RNA editing. Then trans splicing is also there. So in trans splicing, discovered in a few eukaryotic organisms like single-celled trypanosomes, exons from two separate RNA transcripts are spliced together to form a mature mRNA molecule. In trypanosomes, a single exon is sliced into 5' end of any different RNA transcript produced by the cell. In this way, all of the products of trans splicing have the same 5' exon and different 3' exons. The reason that a few organisms use trans splicing is not trans splicing. The slRNA, spliced lead RNA, provides an exon that is connected to the first exon of the mRNA by trans splicing. The reaction involves the same interactions as nuclear cis splicing, but generates a Y-shaped RNA instead of a lariat. Then, in trans splicing, two exons initially found in two separate RNA molecules are spliced together into a single messenger RNA. The chemistry of this reaction is the same as that of standard splicing reaction described previously. The only difference is that another product, the lariat, is the standard reaction. Is the trans splicing a Y-shaped branch structure instead? This is because the initial reaction brings together two RNA molecules rather than forming a loop within a single molecule. Then self-splicing introns. Group 1 and group 2 introns are two distinct classes of self-splicing RNA molecules that contain their own active site for intron removal and exon ligation. These two classes of ribozymes are distinguished by their mechanisms of splicing and by their unique characters. Introns are two distinct classes of self-splicing RNA molecules that contain their own active site for intron removal and exon ligation. Both classes of self-splicing introns perform two constitutive transesterification reactions in the process of exon ligation. The first step of splicing in the group 1 exon involved neophilic attack at the 5' splice site by the 3' OH of the exogenous or free G nucleotide or nucleoside. This reaction adds the guanosine into the 5' end of the intron and releases the 5' exon. So group 1 splicing, first step, second step, group 2 splicing, then third step. The two major classes of self-splicing intron sequences can be readily distinguished in this way. Group 1 intron sequences begin the splicing reaction by binding a G nucleotide to the intron sequence. The G is thereby activated to form the attacking group that will break the first of the phosphodiester bonds cleaved during splicing, the bond at the 5' splice site. In group 2 intron sequences, a specifically active A residue in the intron sequence is the attacking group and the lariat intermediate is generated. Otherwise, the reaction pathways for the two types of sequences are the same. The second step is analogous to the reverse of the first step, following a conformational rearrangement of this active site in which the exogenous guanosine is replaced by the G nucleotide at the 3' terminus of the intron. The 5' exon attacks the 3' exon boundary. This releases the intron and ligates the 5' and 3' exons, no variant formation, but the first and second steps in the splicing reactions are fully reversible because no net energy is consumed. Unlike the group 1 introns, a group 2 intron utilizes an internal nucleophile for the first step of splicing. The 2OH of highly conserved bulged nucleotide of the intron attacks the 5' splice site. This results in release of the 5' exon and formation of a Larian structure whose 5' end of intron is covalently attached to the 2' OH of the bulged DNA. Both group 1 and 2 groups introns are metalloenzymes which require divalent metal cations for activity. Although the metal specificity varies substantially among group 1 introns, it is usually satisfied by Md2+ or M2+. Many group 1 introns in organelle genes contain an open reading frame which codes for a protein called maturase that appears to play a role in splicing. Some group 2 introns are mobile elements that transpose by a remarkable process called retrohoming. In this process of retrohoming, an excised intron comprising single-stranded RNA inserts directly into an organelle genome prior to being copied into dsDNA. Then RNA editing. RNA editing can best define as changing the nucleotide sequence of RNA so that of a mature RNA differs from that encoded by the genome sequence. In eukaryotes, RNA editing is widespread, occurring in organisms as diverse as yeast and humans. Many different classes of RNA editing is transfer RNA, ribosomal RNA, and messenger RNA are edited by various extents. Then site-specific base modification editing or substitution editing. This type of editing include nucleotide transitions or transversions in all classes of RNA. Two very common deamination-based RNA editing is known as C to U editing and A to I editing. However, transamination can also occur as in the case of U to C editing in the Wilms tumor gene. A notable example of C to U editing occurs with the human mRNA for apolipoprotein B. The gene for this protein codes for a 4,5,6,3 amino acid polypeptide called apolipoprotein B100, which is synthesized in liver cells and secreted into the bloodstream, where it transports lipids around the body. A related protein, apolipoprotein 4B48, is made of intestinal cells. This protein is only 2153 amino acids in length and is synthesized from the edited version of the mRNA of the full-length protein. In intestinal cells, this mRNA is modified by deamination of a cytosine catalyzed by cytine deaminase, converting this into uracil. This changes a CAA codon specifying glutamine into a UAA codon, which causes translation to stop, resulting in the truncated protein. In case of A to I editing, deamination of adenosine results in the formation of eosin, which is carried out by the enzyme called adenosine deaminase acting on RNA. Eosin behaves as guanosine. It activates its base pair preferentially to the cytosine, and then present a codon is translated during protein synthesis as if it were G. So pre-mRNA, no editing, site-specific deamination, translation, RNA editing, then insertion, deletion editing. This type of editing is found in the mitochondria of kinetoplastid protozoan parasites, unlike site-specific base modification editing, which occurs at only a few specific sites. The edit kinetoplastid RNAs are very highly modified. In this case, multiple US are inserted into specific region of mRNA after transcription. The first case of editing was described by Benny and co-workers. The sites on the pre-mRNA to be edited are defined by small RNAs that are complementary to editing RNA sequences. These are commonly referred to as guide RNA. The guide RNA have three domains: the 5' region, which is complementary to the substrate pre-mRNA; the central domain, which contains the information necessary to insert or delete uridinates in the pre-mRNA to make the edited sequence and is normally around 30 to 40 nucleotides in length, and at the 3' end of the guide RNA, which is characterized by polyUTIL, the editing reaction takes place in a large ribonucleoprotein complex that has been termed the editosome. Then processing of pre-rRNA. In eukaryotes, there are 4 rRNAs. One of this 5S rRNA is transcribed to RNA polymerase 3 and transcribed by RNA polymerase 3 and does not undergo processing. The remaining 3 are transcribed by RNA polymerase 1 from a single unit, producing a pre-rRNA, which is processed by cutting, chemical modification, and end trimming. Processing and modification of rRNA require a class of small RNAs called snRNA, small nuclear RNA. snRNA base pair with the rRNA to create a disease region that is recognized as a substrate for methylation. Methylation occurs within the region of complementarity. Each methylation event is specified by a different snRNA. A series of enzymatic cleavage produces the 18S, 5.8S, and 28S rRNAs. Chemical modification, methylation, and isomerization, degraded regions of nucleotide sequence, cleavage, incorporated into small ribosomal subunit, incorporated into large ribosomal subunit. Then in prokaryotes, the chemical modification and nucleolytic processing of a eukaryotic 45S precursor rRNA molecule into 3 separate ribosomal rRNAs. In prokaryotes, processing of pre-rRNA involved cleavage, trimming, and methylation of 30S pre-rRNA. There is difference in the organization of the precursor in bacteria. 30S pre-rRNA also contains the 5S rRNA and 1 or 2 tRNA between the 16S and 23S rRNA sequences. Methylation, cleavage, processing of tRNA transcript. Next is about processing of tRNA, which will be covered with the help of next video. Thank you for listening.